So it's 12:45, and it's my great pleasure to welcome you to this lecture. Yeah. So my name is Túlio de Oliveira. I am a professor at Stellenbosch University. I am the director of the Center for Epidemic Response and Innovation. And quite a few months ago, I took also the position of Pro-Vice Chancellor for Business, Industry, and Foundation Partnerships. Yeah, which one of the visions of the Pro-Vice Chancellors, yeah, it is to be ambassadors of the university, and to open the university to outside people. And especially in my portfolio that has to do with both business, industry, and foundations, yeah, the idea is that we make the university quite open, that we welcome business, we welcome industry, welcome foundations, but we also get used to move across the campus, yeah, to each other faculty, each other departments, because we know that is through multidisciplinary and transdisciplinary research that big innovations happening. Yeah. So just before I give to the other Pro-Vice Chancellors to introduce the speaker, yeah, Professor Vikas, yeah, which is the Provost of Efficiency. He's a very efficient man, yeah. So just to ask you a few questions, yeah. How many of you are not from work, current, or study at Stellenbosch University? Okay. Okay, quite a lot, quite like 20, 30%. And how many of you are not from the engineering faculty? Okay. And how many of you before this lecture have walked across the division between faculties and came to an engineer faculty for a lecture? Okay. So that's one of the big objectives of this Pro-VC open lectures, yeah, is that we get used between moving across the campus, moving across disciplines, welcome people from outside the university, because that's where we think that the big impact will have. So thank you very much for coming. Just to let you know that we are likely to get a lot of more people coming in the next 10 minutes, because engineer finished the lecture at 10 to 12 to 1. So in case people is coming. And after the lecture, we're going to have some refreshments outside. So let me invite Vikas to then introduce the speaker. Thank you.
Yeah, thank you, Túlio, and also thank you, or welcome to everybody who is here this afternoon. It's really a privilege to have you. So I'm going to introduce our speaker, which is well known in our faculty. And it's a privilege to introduce Professor Pieter van Rooyen, who's the co-founder of xTenure and in the Department of Electrical and Electronic Engineering at Stellenbosch University. Professor van Rooyen began his career as an academic in electrical engineering, not at Stellenbosch, but at another university up north, before becoming a highly successful technology entrepreneur and life science innovator. He has founded and led a number of venture-backed technology companies across fields ranging from semiconductors and wireless communication to healthcare, genomics, image processing, and the circular economy. Among these was Genome, whose groundbreaking Dragon genomic data analysis platform led to the company's acquisition by Illumina in 2018. His remarkable record includes more than, I hope this is right, Peter, but it's phenomenal, 500 patents and over 50 peer-reviewed scientific papers, hundreds of millions raised in venture capital and equity financing, and the creation of substantial value through company growth and acquisitions. He has also received the Ernst & Young Entrepreneur of the Year Award for Technology. Professor van Rooyen combines deep technical insight with an exceptional ability to translate bold ideas into technologies with real-world impact. Please join me in welcome Professor van Rooyen. And we're looking forward to listen to you, Pieter. And thank you very much for being here again.
Thank you very much for the introduction. Can you hear me there in the back? Is it all good? Okay. Well, so what's interesting about what we do is, indeed, as Professor Tulio said, it's sort of cross-disciplinary. So it's engineering combined with health science and medicine, and maybe we discover some other things that it's related to as well. But what I will be talking about is specifically about cancer and how we can detect cancer earlier. And, specifically, the way that cancer is being detected today is using technology called sequencing. Because if we talk about cancer, we talk about DNA, really, because cancer is a genetic disease. It's your cells that, and the DNA in your cells that mutates and then forms tumors, and then, of course, all the different diseases that follow from that. And I think the best way to explain how DNA works and the interaction to all of this is quite often in popular literature, you read that DNA is a blueprint. So in other words, the suggestion there is the way that the DNA that you're born with sort of determines your path. But it's actually a lot more complicated than that. And a good example of that is if you look at twins, for instance. They have exactly the same DNA, but their fingerprints are different. And the question is, why is it like that? Well, so in the uterus, the two cells that develops is in different locations. So the nutritional gradient is a little bit different. The pressure is a little bit different. And those small environmental changes, and that's sort of the key to sort of the premise of what we do at xTenure. The environmental path that you walk, the trajectory, plays a very significant role in the phenotypical outcome of who you are and also how cancer manifests itself. Another good example is, for instance, the cells. The DNA that you have is identical in every single cell in your body. But if you look at your fingertip, clearly the phenotype that's expressed there is quite different from what it is, let's say, if it's an eye cell. So, and the difference is governed by the environment that it's in. So in the same way, if you walk and there's a little bit of warm air that flows across you, the things that you see, the things that you hear, all those small little changes accumulate over time, and it puts you on a specific trajectory, a specific path in your life. And the best way to illustrate that is if you think of something like a termite's nest. It's not like each termite in the nest knows exactly that this is the plan, what we want to build and develop. They don't have a blueprint in mind that we're going to build this termite's nest that looks like a cathedral. And in fact, you can take that exact same termites that built the one nest and put it just a meter away, and it will build something completely different. And the reason for that is just because the environment is completely different. So everything is based on these small inputs and changes and influences that determine who you are and where you end up. And it's exactly the same with cancer as well. So if we can understand all these influences that precedes a tumor, for instance, how you breathe, what you eat, of course, exercise, we all hear about all these things, but there's a lot of other things that contribute to that too. Infections, inflammation, where you live, the air that you breathe, all those kinds of things. It all contributes to trajectories that lead to a specific outcome, sort of a macro state outcome of where you are. So what we're trying to do at xTenure is to understand what these trajectories are. We want to understand, or we determine all the latent diseases, the diseases that's leading up to the cancer and the tumor that eventually develops. And that's quite revolutionary and the reason why we can do that to a large extent is because of the new tools that's available nowadays. So there's AI tools that's available that can find correlations between different events that was not possible in the past. So if you can keep in mind this trajectory and a personalized trajectory, that's sort of the essence of the technology that we're developing. So, and we determine these trajectories from your health information. So you already have all that information. And we can utilize that to sort of see where you are on your path to different diseases. And we specifically focusing on early cancer screening. So in other words, cancer outcomes. So just a little bit of background on cancer specifically. So I have 2 slides on that. So I don't want to spend too much time on that, but I think it's good to understand the context. So clearly, if there's a personalized trajectory that you're on, the demographic within which you operate plays a significant role. And you can see it clearly in the way that of cancer cases, for instance. It's different, the percentages in South Africa than it is, for instance, in the rest of the world or in the US. The deaths are also not correlated necessarily. Certain cancers are more deadly than others, even though the prevalence might be lower. And another very important thing about cancer is that it's actually a low prevalence disease. So only 1% of people on average really develop cancer. So, and that makes screening for cancer extremely difficult. So let's say, for instance, you have a population of 60 million people, but the cancer incidence is only 1%. So you cannot screen everybody because, I mean, the vast majority, 99% of what you're screening for will be negative. So you need to have some other mechanism, more clever mechanism and a personalized mechanism to be able to detect cancer in an effective way. Another very important aspect of cancer is the stage that you detect cancer in. Stage 1 cancer is pretty much a completely different disease from a stage 4 cancer. If you, for instance, pancreatic cancer detected early, there's almost a 50-50 chance of having a 5-year survival rate versus if it is detected too late or later on. Once it metastasizes, you know, it's really very difficult to detect that. So the key, actually, to curing cancer is early detection. So if you can detect it early, and you can detect it even before there's a tumor, that's really where you want to focus. And that's sort of what we're setting out to do. And I'll show you some of the results that we've done and we've gathered from that. Pancreatic cancer is in particular a very important one because there's only 1 in 23,000 people that gets diagnosed with pancreatic cancer. But if you have it, the chances of survival is pretty low. So again, all these kinds of statistics prove that what you want to do is increase the prevalence, or at least increase the number of people that you screen. And to do that, the sooner you can do it, the more effective the whole mechanism is, and also the treatment that goes with it. So how is cancer being screened today? So it's still the vast majority of cancers are screened with, I would say, older technologies like mammograms. PSA is one that's being done with a blood test. There is CT scans, MRIs, and all those kinds of technologies that's being used, but they're all expensive and the specificity is not very good. The new way of testing for cancer is to look at the cell-free DNA of the cancer cell. So cancer cells mutate and then becomes, or normal cells mutate and it becomes cancer cells, and those cells die, and the DNA of those cells are then in your blood. So from a blood draw, you can then isolate the cell-free DNA, send it to Tulio, and he will sequence it, and you can have some understanding of exactly what kind of cancer you have and from which organ it comes from. And this is where methylation sequencing comes in, because you can actually identify the organ where the tumor originated. So blood draws and isolating the DNA is a great way of doing early cancer detection. There are quite a number of companies that's doing that. Now, the challenge with all of this is really this chart. So you can see to the right-hand side, there are all these technologies that's using sequencing as a technology, methylation sequencing, whole genome sequencing, whole exome sequencing, and all sorts of different modalities of omics. Those are very expensive. The cheaper side is if you just look at proteins, but the specificity and the sensitivity is not that high in those cases. So what do we do in a country like South Africa? If you look at the expenditure of medical expenditure in a country like South Africa, one Gallery test, which is a methylation test, is about $1,000. You can see that the total spend that the government spend on all medical care is about $330. So clearly that's not a very viable technology or way to go about to do screening in a population, not just in developing countries, but also in the US and in Europe and other places like that. So what we set out to do is thinking about ways to be able to do screening, population-based screening, cancer-based screening in settings where finances are more limited and budgets are more limited, and also make it more widespread in places like the US and Japan and Europe, etc. So, promise this is the last equation, but if you look at the way that to calculate this, the accuracy of a test, there are really four parameters that's really important. One is if you do different modalities of testing, let's say, for instance, you look at methylation, look at whole genome sequencing, look at RNA, all the different modalities that you can get from a sequencing perspective. The key requirement there is that all these different modalities that you use for it to be orthogonal. So what does that mean? It means that it's not correlated, so that you have different views on the same condition. And a lot of these tests, this is a test of one of the companies that's developing one of these tests. You can see that 4 of these modalities really is equivalent to roughly 1.8 tests. So it's not very effective. So you pay a lot of money for things that already is not really utilized very well. So a better way to do that, of course, is to find some modalities that's independent, that's not correlated. And this is sort of the essence of what we're doing, and get more into more detail of that. But what we do is we look at health records. So in other words, every time that you go to the doctor, you get an ICD-10 code, a billing code that's associated with a disease that you went to the doctor for. So that gives one view. And from that, we can determine the trajectory of the disease that you're busy developing. Then we do an additional test, which is an immunoassay that sort of makes the that prevalence number higher. In other words, you focus basically more on the people that is at high risk. And then at the end, you do one of these expensive sequencing tests. But the thing is, those sequencing tests are then done in a much smaller population. So as a result, it's much cheaper. But the other upside of that is that the accuracy is actually significantly better as well. So the measure that's being used to determine if a test is a good accuracy is called PPV. it's a positive predictor value. So in other words, how good is this test at predicting that you're busy developing a specific disease or cancer? And the three parameters that's important there is sensitivity. And if you look at sensitivity, instead of just optimizing the sensitivity, you can move the sensitivity from 30% to 90%. And then the PPV changes by roughly 20%. So that's one way, and a lot of the companies that's developing these tests focus, of course, on the sensitivity and also the specificity. But you can see specificity is specifically, if you lower that by just a small fraction, it has a massive impact on the positive predictor value. So the parameter that most companies are not optimizing, and where we see the opportunity is optimizing the prevalence. So you can see if you move the prevalence from 1% to 10%, the PPV shoots up. So that's a very sensible parameter to optimize. And that's where we're focusing on our efforts, is to basically optimize the prevalence and get the prevalence, the risk, the high-risk people more to the front and to be able to screen those people. So the problem, really, if you want to restate that, is not that screening is expensive, but that screening is expensive, untimed, and very specifically unpersonalized. Because if you can personalize it and you know that this specific person's trajectory is on the way to developing a specific disease, cancer, then you can test that group of people and make sure that you can do it more cost effectively. And the way that we do that is really with 5 different levers and 5 different parameters. One is personalization, as I mentioned, to know what your baseline is compared to the population average. Looking at the accuracy, again, as I mentioned, orthogonal evidence for a specific disease that's being developed. Then the cost, how we optimize that is we take it from looking at your medical record, doing a cheap test, and sort of increasing the concentration, the number of the high-risk people, and looking at those people specifically. So we stagger, we don't do all the tests at the same time. We stagger them over time with low-cost tests up front. And then, of course, the very important thing is, depending on what your condition is, maybe you're sick one day, maybe you don't feel that great, and then you do a test and then it's positive. And the problem with that is, if you test positive, there's huge consequences to that. You go for workups, you go for MRI screens, there's a huge cost associated with it. So it's not just the cost of the test that's important, but it's also important to reduce the false positives. And any oncologist will tell you, if you start talking about any screening protocol, they get very nervous about all of this stuff, because it's very quick that the whole system gets overwhelmed, and they get lots of people that should not be there with them, and that shouldn't be screened. So these are sort of 4 of the important criteria, and then specifically also having a gating function to make sure that people that do get tested and they're positive, that you know what the characteristics is of the person. And that's almost a talk on its own, but it's basically to try and understand what your condition is, and to understand if the test that you do, if that's a positive or a negative, a real positive or a negative test. So interestingly enough, the priors that's being used today for all these early cancer screening tests is only 4 things, really. It's gender, age, BMI, and if you're a smoker or a non-smoker. So those are the priors that go into all these tests when they do clinical trials. And you will see our approach is definitely using your personalized information, your personal trajectories, where you are in your personal path and road, to be able to use that to determine if you should do a test or not. So this is an example of a specific person. And this is, over time, over age. And you can see this person has developed sort of 5 diseases that is unrelated to cancer over time. And at a certain point, that person's probability of developing pancreatic cancer has risen to 41%. And if you compare that to this bottom curve over here, which is the population, if you use the population as a measure to understand if that person is at high risk, you can see that you clearly would have missed it. So we have an AI model that we trained. And from this AI model, we can then take a person's health information and labs that the person has done. And then from there, determine all these diseases and the trajectory of all these diseases to determine cancer risk. And the test that we are working on and developing is this personalized cancer risk trajectory. I will give you some more information there, but apart from, instead of it being just a test, it's actually more like a system. So it's a continuous system that updates and gives you sort of a continuous updating of where you are in your risk profile. So first run this on your healthcare data. Then if you're low risk, of course, that you just go back into the queue and, over time accumulate your specific data and determine a risk profile from there. And if you're high risk, we do an immunoassay panel. You can recall maybe from that earlier slide, the immunopanel assays is about $25. That's the cost price, so it's significantly cheaper than an expensive $1,000 test. And so there's like already 2 components in which the risk is being determined. One is from your medical record and one is from your immunoassay panel. Then you continue to the next step, and there's a gating function that determines if you should take another test before you go for a workup or expensive test. And if you're low risk after that, or if you're still high risk after that, then you do a test, let's say like this methylation test, the Gallery test that's using sequencing. And so one of the potential collaborations here with Professor Tulio is to have a test like this here, like a DNA-based test here. So then the whole cycle of our lattice test can be really done locally, which I think would be really, very big advantage. So, and the types of cancers that we can detect is quite extensive. You can go from liver, ovary, all the way to prostate cancer. And all these different cancers, of course, have different outcomes and different probabilities of success. But on average, if you compare it to some of the expensive tests, doing it in this sort of iterative system-based way, in a low-cost way, with an expensive test at the end, you can basically get a very effective test that's equal to the most expensive test that's being done that's only sequencing-based today. So what does the numbers really look like? So the key, as I mentioned, is to increase the prevalence, to increase the number of people that you test and identify the number of people that you test. So in a population of, let's call it 100,000 people, the prevalence is 1% for cancer. So there's 1,000 true positives in there somewhere. So out of the 100,000, you need to figure out which 1,000 people in the 100,000 is actually positive. So first thing is to look at the medical records, and from there you get about 400 true positives and a huge amount of false positives. And that's the number that any oncologist wants to run away from. So you cannot just do the medical records. So there's after the test, the prevalence goes up to about 3.9%. Then after you do the on the high risk people, you do an immunoassay panel, and then this happens. I don't know what happened now. Yeah.
Are you still on Teams? Yeah, it would have helped a lot if we had electrical. I'm mechanical, right? So I can omit slaan.
Maybe unplug it in some way.
It might have lost you.
That's the engineering solution. Unplug it.
You're going to share again. Did you use Apple Cast or something fancy like that?
It's not
No,
that.
he's just on Teams.
Oh,
Yeah,
okay.
that's
Otherwise, we can just plug it in. Then it's possible.
for the
So we
hybrid.
just need it to show. Sorry about that.
No, it happens.
It's coming.
Maybe you want to
Yeah.
plug it in.
If you plug it in, it will show there.
Oh, there we go. Excellent. Thank you.
Yeah.
Yeah. Thank you. So that was a short break. Yeah, so just to recap, so we do the personalized test, get the personal trajectory, then do the immunoassay, and then we increase in 2 stages the prevalence to 13.5%. So, and if you can recall from the first slides, if you increase the prevalence, the accuracy of the test increases quite significantly. So in the end, you have a very small number of false positives, 62 people out of 100,000, which is 0.02% or something like that. It's a very, very small percentage of people that tested positive, but that's not positive. The number of true positives is about 190. Now, clearly, that's lower than 1,000 than expected. But this is where the gating function comes in, because those people are not cleared. They just go back into the queue again so that they can be sort of flagged and tested again over time. But the end result of this is really on this. This is sort of the most important thing, is that if you were to screen 100,000 people in a population, it will cost, if you just use, let's say, a Grail test or a more expensive DNA-based test, you will basically have to spend $280 million. And remember, you have to do this every year to screen a population. So clearly, that's nowhere in the world that is possible. So it's about $331,000 per cancer. Our approach, the lattice approach, costs about $2.2 million. And the important thing is it's about $23 per person over the whole population. So it reduces the cost and the access and the feasibility to something like early cancer screening test, by orders of magnitude. So, and that's why we're very excited about this and this approach, because we think it can really have a significant impact and make a big difference, not just to a South African population, but it really can be proven here. Because if you think about it, if it works here, you can obviously scale it very effectively to any other other area in the world as well. So that sounds all good, but does it really work, right? So what we've done is we've used 500,000 UK Biobank patients, their data. And the UK Biobank is a data repository where there's all the health information of British people that's in the British health system. They have an NHS, their National Health Service, so it's centralized. And we also have the cancer outcomes for each one of these patients. So we've taken all these patients, we've listed all the diseases that's relevant, sort of apart from rare diseases. It's 1,250 diseases. And we've mapped, trained our AI model to be able to, from the data, determine or map the patients to all these different diseases so that we have a roadmap, if you will, of the diseases that's leading to cancer. And the mechanism is something along these lines. So firstly, obviously, people have to give consent. And this is also how it will work in practice, you know, if you want to implement this. You look at all the ICD-10 claims, the demographic data, blood markers, so all the data that's in the healthcare system about a specific patient. Then we've trained the model on 400,000 people. And then validated it on an additional 100,000 people. So this obviously can be extended, and we're working with companies to extend the number of people that we're using this to train the model and make it broader. And important about this is, you know, obviously in a specific demographic, it needs to be trained on people in that demographic. So in other words, if we want to do it in South Africa, we need 3-4 million plus people to train the model to be accurate for this demographic, and then we can use it with confidence on people in a specific demographic. But what we do from this is then we map the diseases onto the outcomes, the different cancer outcomes, and then from there we can actually look at the history to do the training, and then we can project it to the future to do projections and predictions of where this person's health is going. And what it looks like is something like this. And this is sort of a scatter plot of all the different diseases and their relation to each other. So the bigger the dot, the more of these diseases are present. And if they're close to each other, they're more related to each other. And you can see in this particular case, we've mapped the different regions where cancer occurs. So there's about 14 different regions. And you can see on the left-hand side here, the pink, those are all pregnancy and childbirth related diseases. So they sort of clustered independent from all the other diseases. So, but now what's interesting, if you were to look at this and use the traditional way of thinking, you would say on average, this is a good way to do predictions. But it's actually highly dependent on the person. And one person developing, let's say, pancreatic cancer has a different map than another person. And that's where personalization really comes in and why it's so extremely important, because each person is different. It's like that termite's nest. You know, each person has its own nest that is building, but it's your own path that determines what your disease profile looks like. And that's what we're determining, is what is your profile, and from there determine what your risk is. So this is a good example of exactly what one person's trajectory looks like. And if you look at the map, the disease map, you can see the black spots is all the different areas where this person's risk for pancreatic cancer came from. And you can look at the trajectory over time, and you can see at age 65 or 68, there's a big increase in risk to develop pancreatic cancer. But this person's risk looks quite different from this person's risk. If you look at the map again, it's a different distribution of diseases that's leading to the same outcome. And what's important about this, you have all these different diseases, and now you can develop therapies. Instead of treating the cancer, you can actually develop therapies to address the diseases and have interventions on the diseases before the cancer actually happens. And we believe that this could be really revolutionary. It's much cheaper, so you can really deploy it at a population scale and have a big impact in that way. But I have to add this one. There's more. How do you give everybody access to technologies like this? So firstly, people obviously need to consent to giving their medical history and information. And then what do you do with this immunoassay, this test that you need to do? Well, I'm a co-founder of a company called Truvian Biosciences there in San Diego. And we have developed a desktop analyzer where you can basically look at all the traditional run-of-the-mill tests, like complete blood count, cardiovascular disease, liver panel, kidney panel, all the tests that you do when you go to the doctor, for your regular checkup. So it does all of that in any sort of location, like here in South Africa. It could be in a disc game, could be at health clubs, etc. So it takes a small amount of blood, put it on the disc, and within 30 minutes, you have a complete panel. So that's sort of the wellness panel, sort of the panel that everybody does every year or 18 months or 2 years. So what we're busy developing together with some of our partners in the space is to convert or deploy this immunoassay onto this device as well, so that you can basically then have easy access to all these devices and then basically determine if you're at risk and where you are on your trajectory together with your health information. So that's another thing, if you can remember during the COVID times, one of the problems was where do you go for a test, right? So it's one of the problems that this device solved, because devices everywhere, you can develop new tests on the device and have feedback on diseases like infectious diseases, but in this case also have an early cancer screening device. So it's sort of unprecedented, because for a very small amount of money, let's say $60, you can do a yearly cancer screening test. It makes it accessible to everybody in the population to be able to do something like this. So we're not doing this on our own. We have some partners, and some of the partners that we have is Synexa. Justin there, he's going to develop the immunoassay. Task is helping us on the clinical trial side. Stone 3 has been always great supporters and partners. They've helped us to get all of this up and going. We have UTF as a fund, and so what we would like to do is extend, an invitation to discuss with anybody that has ideas, research groups, etc., to help make this really something that can be an early cancer screening test that can be developed in South Africa, have an impact here, but also can be exported to the rest of the world in a very cost-effective way. So that's basically my story. So thank you very much for being here. We're excited to have this, and hopefully we can deepen the collaboration with anybody that has interest. Thank you very much. So if there are any questions.
No, no, no. It should have a lot of questions. One thing that we do well in our lectures, yeah, is ask lots of questions and gently grill the speaker. Yeah. So please, questions, comments.
Peter, thank you very much. So, Wim de Villiers, and I have to declare a conflict of interest, and that's that I'm a gastroenterologist. So I have a very strong interest in colorectal cancer and pancreatic cancer and liver cancer. And one of the big tests that gastroenterologists do is screening colonoscopy, where we try and screen and prevent the development of colorectal cancer. And the sort of run-of-the-mill answer there is if you have, we would expect about 70% of our screening colonoscopies to be negative and only about 25 to 30% to be positive, and that would require, and that's detection of a polyp, which is a precursor of colorectal cancer. It doesn't mean that it is cancer. So very interesting. What you're basically describing, it's all math. All right, and you're enriching your sample, your
Population that you do.
yes. So, but it's also very dependent on the ICD-10 code. So it's dependent on the correct information in the healthcare system, which is a potential problem, which is a potential issue in terms of not only in the States where I worked for a long time, but definitely in the South African system where the coding may be problematic, and also access to an electronic healthcare record and or previous records. So, and then you enrich the sample, and then you have an immunoassay as your first line test, which is about seven, I think it's seven, is it?
7 markers.
7 markers there, which is fairly limited, and some immunoassays we know are not very specific for cancer. But so the issue is how is again it's garbage in, garbage out. So if the ICD-10 code is inaccurate and it's garbage, then it could be a significant problem.
Yeah. So we've looked at that, and of course, you know, if it's complete garbage, then of course that cannot be used. But it's not only ICD-10 data that's been, and that's where this whole different orthogonal modalities come from. So labs is one that plays a very significant role in determining that concentration factor. Any data that you have on a person is being used, and the model is trained depending on what the data is, and that's why it will be good to train on South African data. That the model can reject things that doesn't line up with what makes sense. But then on the immunoassay side, at the moment it's 7 proteins, but we will probably extend that to more to have a bigger array of inputs. But what's also important is that immunoassays like PSA is not very specific, but the combination of all these different markers together actually has an outsized importance and impact. So it's not using a one-to-one marker. That's why I have in this slide, it shows sort of a, combination of all of this. All of this together with the healthcare information actually does make a significant difference. And you know, even PSA, which is a low test on its own, does make a significant difference.
Yeah, that's quite a grilling, Wim. Yeah, for you. Yeah, yeah. Just other questions. We have 2.
Hi, thank you so much. It was very interesting. I'm new in the cancer research field. I was just wondering, a lot of the whole backstory of it comes from the history of patients and everything in the records. For interest's sake, what is your plan or idea regarding people like cancers like osteosarcoma, where it's younger people? where you're like the younger cancer prevalences, where you won't have background, you won't have previous diseases.
Yeah, so I think if there are diseases that falls outside of the scope of the training data, then we will obviously not be able to detect that. But the main cancers, like the ones that's listed here, is probably 70 or 80% of the cancers that's currently affecting people. But there will be some that's outside of that, but that's normally even a lower prevalence, right? So it's a very low indication.
So also just with that, in any of the AI predictive models, do you consider relatives, like parents' history with the person's history, or is it just the single person?
Yeah, no. So you're basically talking like in genomics, like
Yes.
PRS, polygenic risk score, and family history. So I have quite a few friends that's doing research in the area, and although it is important, but again, if you think of that sort of termite's nest, the importance of the environment, there's a lot of people that have a predisposition for certain cancers, but never develop it.
Yes.
And there's also people, of course, that do not have a predisposition that develop cancer. So it does play a role, family history and your DNA, but what's outsized really in terms of determining cancer is the environment that you're in. So lifestyle, all those kinds of things. It's really important to be conscious of those things. And I think that's another advantage of this is once we show people the trajectory that they're on, there's also a way then for them to change certain lifestyles to be able to reduce your risk before something like that happens. So I think even for cancers where that's not being addressed in our system, you will still get valuable information out of that to look at how to minimize certain of your risks.
Thank you so much.
Thanks.
I just want to make sure I understand exactly. So it seems to me like in your talk, you've got two things that you're bringing to the table, two new things. Like the one is you're saying, we're going to look at your medical history and we're going to ask the computer what your chances are. And then the chance is high enough, we're going to give you a test. And
yeah, the second test.
And then the second thing is you're going to make a little black box that you're going to put in DisChem that's going to be cheap and it's going to do this test for you.
Yeah.
So like the first thing is not revolutionary. It's an advance. But I mean, it's like we already checked your medical history and we do all that stuff already. The only thing you're bringing to the table is like, okay, I'm going to use AI now as well.
Continue the question.
And then the second thing with the black box that we're going to have in DisChem, that's kind of cool. Yeah, that's all.
So I would say looking at your medical history, right? You do not get a disease map. And specifically, you don't get a trajectory of the diseases, the path that you're on to get to a specific disease. So I think that is definitely novel. So it's not just looking at your medical history as an interesting input to where you are. It's the actual trajectory of it's the actual diseases that lead to a specific cancer. Maybe this is not the best one. It's maybe this one. You see, so for instance, if you look at cancer, for pancreatic cancer, you would say everybody that's diabetic or everybody that's overweight is at higher risk. But it's not necessarily the case because your disease history might not include any one of those. I mean, there are many people that get cancer that's not overweight and that's not diabetic. So it's the actual diseases that lead to a specific cancer and a specific disease that we determine from the model. And I don't think that is something, if you go to a GP, that you just get from them. And I think that's what's novel about this. We look at the trajectory, the personalized trajectory, and what the chances are of you developing that specific disease. And then the black box, I think that's relatively revolutionary too, because if you go to, you have to go to a doctor to get a blood draw. it's a way higher barrier to entry than if you can just, every time that you go to a dischem or whatever, get that test done. So I think it's revolutionary in that sense. And it's also the cost. I mean, how do you screen for cancer? You have to, today, the only way to screen for specific cancer is you have to go and get a very expensive test. And what we've shown here is using these simple, low-cost methods, we can get sensitivity and specificity and positive predictive value that's better than most of these tests.
Hello, Pieter. So I'm Dr. Edberger, Head of Oncology at Tygerberg Hospital, Stellenbosch University. So this is very interesting to us, and I am that person that is very scared that you're going to diagnose all these people. In fact, I'm not. It's the surgeon who's going to be very scared because he's going to have to do the scopes and biopsies. Pieter, I want to maybe highlight an element that I want to test with you, because I think I also, for the first time now, I've heard your plans, and for the first time now realized is it true that you're not only looking at risk factors and diseases that might lead to cancer, but you're looking for a pattern in ICD-10 coding, especially the one where you mentioned the ovarian lady. So we say that patients get diagnosed late, and we as doctors like to blame the patient. So we say, oh, but you didn't go. You didn't report to the doctor that you have vaginal bleeding and now you have stage 3 cervical cancer. But there's a very large component, and more studies are showing that the medical system is failing the patient. The classic example is your bladder cancer patient who will tell you, but doctor, I went to the doctor year after year and they treated me for a bladder infection. And so those are the ICD-10 codes. Am I reading this correctly? So you're going to read those misdiagnoses of an actual early cancer developing, like the lady with the ovarian cancer that was treated for gastro. That was never gastro. That was early ovarian cancer, but it was misdiagnosed.
Yeah, that's true, but that's why labs as well is part of it.
Yeah.
And all the other information that's in the system. So ICD-10 codes is one part of it. And yes, I agree. There will be obviously some representations and errors in there. But despite that, and our model shows that if you look at all the data that's present, you still get a very good indication on the outcomes of an indication of what kind of cancer it's currently in the process of development.
Okay, because that would be really great, and especially in the state sector where you see a different doctor every time, or where you go to a different GP every time, that the pattern is not recognized. And then the other thing is that we also need to understand is that this can only pick up cancer that is already established, and usually only when it's stage 2 or higher. Especially stage 3 and 4. And so this all happens before any diagnosis or treatment.
Yeah.
So the cost of diagnosis and treatment will still remain the same, and it will actually increase because we are now inputting more people into the system.
Well, but the thing is also you will probably get more of the right people, right? So people that actually are true positives.
Yeah.
And as you can see, if you go through the whole funnel, that the number of false positives are actually reduced quite significantly.
Yeah, but what I'm saying is we're currently only treating people that really have cancer.
That already has cancer.
So we're going to add to that load.
Yeah, you probably will, but you would have seen them anyway at some point.
Yeah, just later. Yeah.
But the thing is, I think what's important about this, you will see them at a point where you can actually treat them, as opposed to a point where it's a lot more of a burden on you than
on the surgeon.
Than it would have been if it was early on.
And then my last thing, I will stop talking now, is the psychosocial side of this, where we in our country, maybe in the private sector, the 20% private sector population, it's a little bit different. But in the majority of patients, they don't want to know that they have cancer. They're actually not that interested, most people. I'm talking about the average person. And so I would be interested to know what, let's say, parallel processes do we add to a project like this, for instance, in educating patients about cancer symptoms, educating healthcare staff, and actually making people willing to be diagnosed with a cancer and helping them understand that it's actually to your benefit. So how do we change the public's view to actually volunteer to be diagnosed with cancer?
Yeah, well, and I think this is sort of the invitation in the end, you know, working with people like you that understand those issues and trying to address that in a broader way, not just through testing.
Yeah, I can see Wim and Richard, but I'm not just the microphone carrier. Yeah, so I also have a few questions. Yeah, for people that don't know us, the CERI, yeah, we host which is the largest and most automated genomics, this means DNA sequencing machines in the African continent. Yeah, we have been thinking a lot about that. One of the things that we decided early, and we are very happy to reconsider now with your results, is that at the moment the test that he defined, the expensive one, that the name is that's the methylation, the Galleri, it produces a lot of true positives, but a lot of false positives, and people end up only using this test now when they don't need that. People that are already sick with cancer, then they spend money that they don't need. Yeah. But what I like about your algorithm, yeah, is that basically it gives a timing when to use this expensive testing, yeah, which then make very cost effective for the health system. I can clearly see the medical aids like Discovery or some of the big medical aids being very interested on that, because that would save quite a lot of money for them, which would make people a little bit more healthier and higher survival. Yeah. So I can see very strong utility, and we are very happy to discuss further how we can work together, because I think that cancer is going to become a big problem in South Africa. Our population's aging, yeah, and we also have a lot of comorbidities. Yeah, in relationship to the IC10, yeah, Wim, is not a difficult thing to solve, because the discoveries, the private medical aid will have that. The NHLS also have that, because they really control things. And you can also do, if you don't have that information, you can ask a questionnaire, and then before, yeah, with the patient, and then add that manually. But one question, without, I'm trying to be very positive about your thing, which I am. Yeah, it is. Okay, given that you are waiting, so you are there. The person come and you do the basic, the medical analysis, yeah, and then you wait, and then you do your immune assay, then you repeat, and only that you're gonna do the methylation test, the Galleri. But in that process, you are not also reducing the survival at 5 years, because you are detecting a little bit late, or not the case in the specific patient. I'm not talking about the population.
Well, so just repeat the question. What exactly?
So you remember one of your first graphics that you show is the survival rate by the early detection. Yeah, and it's quite a lot for breast cancer. It was close to 100%. Yeah. And but by doing that test, yeah, have you checked the survival rate? If that decrease, it should decrease a little bit, because potentially you're detecting a little bit later, but how much would that?
Yeah, no, so I mean, we still need to do clinical trials to show that. But the thing is, if you can increase the PPV, the positive predictor value, it will definitely have an impact on the survival rate, because if you can detect it early and you can predict that you're going to get it, then you can do something about it. But it's not, we need to still do a clinical trial that's in that regard. Yeah.
Okay.
Hey, Peter. So I really enjoyed the talk. Thank you. And I think the move to prevention or early detection, I think, is definitely the future, rather than treating people with very toxic drugs when catching them late. And I think the health system challenges is what we're all really talking about. I understand why you chose cancer, because that's where the money is at the moment. How well do you think this would work if you were to do it in diabetes or hypertension?
It's the same principle, so you can basically determine the diseases, and we have some models and results on that as well. So I don't think we chose it specifically for the money. I think it's just, you know, I think anybody that's had friends or family that's been impacted by cancer, it's pretty, I think if anybody can choose, they would choose to help somebody with that. So I think the impact of this could be very significant, and something that can make a real difference.
I was informed that we are running out of time, which is good because we have a lot of questions. I'm going to give one question here, and then I'm going to give back to Vinda Villas because his office is next to mine. Otherwise, he will complain when you go back.
Pieter, very interesting as always. Great to see somebody with the courage to tackle hard problems like this. So my question is, what is the most important technical barrier in making progress here? And then perhaps even more interestingly, what is the most important non-technical barrier that you're facing in making progress?
Well, I think access to data is probably the biggest one. To address maybe a lot of the questions here is to really show what the extent of mislabeled ICD-10 codes are. For instance, we've looked at that, but 500,000 patients is not enough, especially in a South African setting. Of course, that will vary as well. So I would say technically that's probably the biggest challenge. You know, and I think maybe what Riet mentioned, you know, the education around testing, because most people care more probably about their car's oil change than about their health until there's something that's really wrong with them. So I think it's the education around that as well, and making people understand that it's an important thing to do, and it's not just important for you, you know, it's sort of important for the community that you live in, etc. So
yeah, thank you, Pieter. And I also want to concur. It was a great talk, and you really did that very well. And I want to come to your defense somewhat in saying that I think the big advance here is the trajectory individualized data from a multiplicity of data points that speaks to personalized medicine, and that is the future. Because at the moment, public health guidelines are, you know, at the age of 45 or 50, you need a colonoscopy, or when you're sexually active, you need a cervical cancer screen, a pap smear, etc., etc., etc. But if you can personalize that, it becomes much more valuable. So I really think that's very interesting. I would also, and it's what Richard said, I think there's a huge scope for this, for example, in non-communicable diseases, in cardiovascular disease prediction, dementia, Alzheimer's, vascular dementia, etc., etc., and also looking at circulating tumor markers and so on. Now, about the black box issue, I have to ask you, and there's a whole host of these type point of care, finger prick type tests, and I just have to utter the feared word Theranos and Elizabeth Holmes, and then one starts to shudder.
well, you can see I don't have a turtleneck on. No, so actually, what the Struvian device is, it actually quite interestingly started in South Africa in 2010. We had a project at the CSIR that I was involved with to look for TB, and TB is actually best detected using sequencing, right? Because you get different strands, etc. But what that device does, it does 32 tests that's FDA approved or cleared, unlike the Theranos device. And it does all the standard testing, CBC, liver function, kidney function, all those kinds of things. And specifically, it is FDA cleared, which is different, and it was just cleared last year. So it's not the same as just a diabetes finger prick kind of device. It has 3 modalities in it. It's got a microscope in for CBC, a fluorescence microscope, and a chemistry analyzer. So it does everything basically apart from molecular testing. So, and it has a disk that you can put with wells that you can add additional features to that, like for instance, these amino acids. So yeah, there's the Theranos story, but this is, think of it as Theranos that works. Okay, thank you.
And Wim, there's a big difference. Now there are South African engineers involved, right? So that makes a big, big difference. My point exactly. All right, colleagues, thank you very much. I think we can clap one more time for our speaker.